All right, let's talk about chlorophyll fluorescence now. So chlorophyll, it's a hard word to say. Don't say it 10 times fast. Chlorophyll fluorescence is a technique for assessing the health or functioning of photosystem 2 in particular. I'm going to talk about why it's just photosystem 2. So it's a little more of a technique than a concept of plant physiology. And usually I don't try to do those types of things or cover those types of things. But there are a couple reasons why I want to do this. I guess we'll jump down to the third bullet point. One, I think as we walk through the process of what of what chlorophyll fluorescence is and what it measures, it helps us understand some of those light reactions in photosynthesis a bit better. And then also, the other reason is that in the last few years, one of the newer satellites, and I don't remember the name now, gets chlorophyll fluorescence. So it can't generate all of the metrics that we're going to talk about today, but it can generate some of them. And so that's going to be a really powerful tool because we can, get chlorophyll fluorescence on a very large scale fairly frequently. And at some point, and even now, it might be completely free to get those metrics. So, if you're thinking about research on a large scale, or you're thinking about managing a particular region, being able to download that data from satellite imagery is going to be a really powerful tool in kind of assessing how particular ecosystems are behaving and functioning under different scenarios. And then, excuse me. Another thing that's important about this metric is it correlates with photosynthesis well. It's not actually measuring CO2 uptake, but the metrics we can get from chlorophyll fluorescence correlate really well with photosynthesis under most conditions, and it's way faster to make these measurements. And the systems to measure them are a lot cheaper. So it's a good alternative if you don't actually need to measure CO2 uptake. Okay, let's talk about what's going on in chlorophyll fluorescence. Once light is absorbed by the antenna in chloroplasts, and so remember we got photosystem 1 and photosystem 2 with those antennas, those different pigments absorbing that light energy. Once that energy is initially absorbed by those pigments, there are three things that can happen to it. It can go on to actually drive photosynthesis, which is this phi sub p. It can be dissipated as heat. So if it doesn't get used in photosynthesis, it might just convert to heat. Or it can be re-emitted as light, which would be at a longer wavelength than what it came in. And that's what chlorophyll fluorescence is. Heat dissipation is this phi sub d, and then fluorescence is phi sub f. And because the wavelength that it emits light during chlorophyll fluorescence is longer than what it absorbs at, we can measure it really easily because we can give a leaf the light it needs for photosynthesis and then look for longer wavelengths coming off of that leaf. And if we see longer wavelengths, then we know that's coming from chlorophyll fluorescence. Just to clarify, all of the fates of life that would be incident on a leaf or hitting a leaf, we, like in this system and with chlorophyll fluorescence, we're talking about these three functions that are on the right side of this figure: photosynthesis, chlorophyll fluorescence, and heat. But some of that light doesn't even make it into the leaf. It would be reflected from the leaf, and then some of that light gets transmitted right through the leaf itself. And we talked about that a little bit during the light reactions lecture, so I just wanted to make sure to highlight all the different paths that light can take once it hits a leaf. Fluorescence is really, or these three fates of light are what happens once it gets absorbed by photosystem 2. Okay, a couple things here. Mainly I want to highlight why we're only measuring photosystem 2. So what we're going to do in fluorescence, a real quick snapshot is we're going to blast it with some light and then look at how much is reflected, and that'll tell us something about the capacity of photosystem 2 to absorb light energy. And the reason it doesn't work on photosystem 2 is because photosystem 2 can receive energy from different places. One, it can get it from light. We know it absorbs light at slightly different wavelengths and longer wavelengths than photosystem 2. But it also can receive electrons from photosystem 2. So it's receiving energy from both places. And what that means is we can't isolate photosystem 2 in the same way we can isolate we can't isolate photosystem 1 like we can photosystem 2. And so we can't really measure it in the same way. So what we're going to be measuring in chlorophyll fluorescence is only about photosystem 2, which that's part of its limitation, is we're not capturing the whole photosynthetic machinery. But in general, whatever is happening to photosystem 2 tends to be happening with these other enzymes that are part of the electron transport chain. So it's a good representation of what's happening. The other thing I just want to point out and talk about here, and we'll come back to this when we look at the actual data from a chlorophyll fluorescence measurement. But when light, is coming in to a photosystem and it excites the electrons in that photosystem, when those electrons are excited, for just a brief fraction of a second, that photosystem can't absorb any more light. Any new light coming in is then going to get released as fluorescence, okay? In that case, when that electron is in an excited state, we would call that photosystem closed because it can't accept any more incoming energy. But then as soon as that electron is passed off and goes down that Z-scheme, then it's open again and it can absorb energy. And that happens like on a fraction of a second. But it comes into play during these measurements. Okay, so I don't know if this helps to see at the beginning or at the very end of this explanation, but here's just an image of a unit, an instrument that measures chlorophyll fluorescence. So it's much smaller than the 6400s that we were using in lab. And you can see there's a light being emitted onto the surface. And then this probe here, it emits the light and also measures the reflectance coming off of that leaf. And it can do this emission and sensing of the fluorescence also on fractions of a second. So it's operating really, really quickly to capture these dynamics of those light reactions. All right, let's ignore that for now. Okay, I'm going to try to draw this again, because for me it helps to think about this as a time series, which it is. So this figure on the right, I'm going to draw basically that figure. And this is a time series of what's happening as a plant is exposed to different light environments. So we'll come back and talk about this figure, but I don't know. I think this might help. It helps me. So you're stuck looking at it. Okay, so what I'll do is we'll draw some axes. So the x-axis is going to be in time, and this whole process is going to take a few seconds. All right, so it's really a short period of time. All right, and then over here we've got fluorescence. Oh, that's where I can't draw past there. All right, so we've got fluorescence. And I'm going to draw 0 right in here, and it doesn't really, the values of fluorescence don't matter, and I'll tell you why in just a second. So I'm going to draw you a picture. Oops. Just a second. All right. Draw a picture of what's going on during a measurement. Okay, so you're gonna clip it on a leaf and start measuring. And so the fluorescence value is gonna be 0 at the beginning. Oh, and I should say one thing that's in the title. This would be referred to as dark adapted photosynthesis. So this is a leaf or a plant that's been in the dark for a while. And so we know photosynthesis is completely shut down, okay? So that initial fluorescence value is going to be 0 because it's in the dark, and so if there's no incoming light, it's not emitting light. At this point, the instrument is going to turn on a very low level light, or I don't know why I call it MB. It's ML. Let's just scratch that out. All right, so this is a very low level light. It's not actually turning on photosynthesis. It's just the light it needs to get some fluorescence back. And so we get some low level of fluorescence happening at this point. All right, so if we look at this real quick, I'm going to draw all of these parameters that we've got here, F sub 0, FV, FM, all of that stuff. We'll come back and talk about, like, they're all defined here. So hang on as I draw this and we'll come back to these. Okay, so this is going to be, this level here is F sub 0. Okay, so we've got a little bit of light just to get some fluorescence. So this is like our baseline fluorescence. This is just kind of the noise we would expect when there's a little bit of light coming into this leaf. And then a fraction of a second later, the instrument is going to provide a very, very short pulse of saturating light. So really bright pulse of light with high intensity. And we're going to see fluorescence spike during that time. That's a horrible spike, but you get it. And this is FM. All right, so like maximum fluorescence. So F sub 0 is the baseline fluorescence, F sub M is the maximum fluorescence. And then what we're going to use as a measure in our calculations is the difference between those two values. Okay, I'm not going to draw it now, but that's going to be F sub V. We'll see it on the next slide. And I drew that a little high, but let's say we've got a spike, and then right after that, I guess this is fine. Right after that spike, we've got another light source that's coming on. And this is referred to as AL. And so this is enough light to drive photosynthesis, but it's not going to be a saturating light. All right, and that stays on for a while. And what happens is you start at relatively high fluorescence when that light is on. And then it slowly, as all the photosynthetic machinery turns on, the Calvin cycle starts moving, all of that kind of stuff, that amount of fluorescence starts to slow down. It starts relatively high because the electrons and the energy that's being produced by the light reactions, they're not being used by rubisco yet. And so it kind of backs up, and that causes fluorescence to be relatively high. But as rubisco starts and the Calvin cycle starts moving, it's using that energy. That opens up the photosystem, those reaction centers. So I talked about them being closed, meaning they can't accept new energy, or being open. This, I should go back and say, this big pulse indicates that those reaction centers are closed. But the size of that peak tells us how many reaction centers there are. So if there's a lot of reaction centers ready to do work once they're open, you get a pretty big pulse. Okay, so as this curve tapers down, those reaction centers are starting to open up more, and we see this kind of more baseline value of it's not baseline, it approaches F sub 0, but more like a steady state is the word I'm looking for. A steady state level of fluorescence. And then further down here, we're going to get another saturating pulse. Of light. And so we'll see another spike. Okay? But this spike is lower than this other spike. Remember F sub M, that maximum rate, that's when photosynthesis is shut down. And so this is kind of just telling us how many photosystems there are. Here, this is referred to as F sub prime, or prime M. I don't know why I called subprime. So it's the maximum fluorescence after photosynthesis is turned on, basically. Okay. And let me make sure. I think there's one more. I need to one more definition. Oh yes. Okay. I don't think we'll use this one, but this level here. So remember, F sub 0 is that baseline. This value, when photosynthesis has kind of like reached this steady state, this is F sub t. All right. So all of these are different levels of fluorescence at different times. The main things we need to know is that the height of that peak is telling us about both how many photosystems there are and how many of them are closed. Okay. And again, this time series I'm drawing happens in like a second. So this is all happening very quickly. So I don't know if that helped or not, but here's this diagram here. So this would be our starting point. Here they've turned on, oh, they do call it MB. We also call it ML. But anyway, so this low level light turns on, brings it up to F sub 0. This is our baseline. Here's that saturating pulse of light. So that's our maximum fluorescence. And again, I mentioned that F sub V is that difference between FM and F0. Then we have the photosynthetic light turn on. We see this pulse in fluorescence, and then that decline as all of the photosynthetic machinery starts to turn on. As we plateau, that's when photosynthesis has reached a steady state. That's F sub t. Then we hit it with another saturating pulse of light, and that gives us F prime max. And so this tells us, this gives us an indication of how many photosystems are open and ready to absorb light when photosynthesis is moving along at kind of a steady state. All right. So the higher this is, the more photosystems that are closed. The lower it is, the more that those photosystems are open and ready to do photosynthesis. And so in this case, this second F sub m, if the photosystems are damaged in some way, we're going to see a much bigger spike. But if the photosystems are working well, we should see a relatively low spike in that particular area. So there's a bunch of different metrics we can get from this measurement. And I'm only going to talk about a couple that are the most common, and I think they're the most useful for the widest range of people. Some of the other ones are like fairly detailed measurements. I'm going to skip all of this because I've talked through them, but there they are. Okay, so one of the main metrics that we use for looking at the photosystem of different plants is FVFM. So let's go back and look at that. So FV is this difference between F0 and FM, and then this is FM. Now in this particular graph, they put this little degree sign up here. Ignore that, we don't always use that one. And so when I say FM, I'm talking about this one without the prime. So FV over F prime. So that tells us the proportion of the light absorbed by chlorophyll that is used for photochemistry. All right. So the larger that number is, the more of the energy is going to photochemistry. And the reason why is if FV is really short, that means most of the photosystems are open and absorbing that light. If FV is really big, it means those reaction centers were closed and they're fluorescing light back off. And if they're closed, that means they're using that light to go through the light reactions and drive photochemistry. If it's really small and they're open, that means they're not actually utilizing that light that we're blasting them with, or it might mean that that light is being dissipated as heat, because we're not measuring that in this particular example. So FVFM tells us what proportion of the light is being used to drive photochemistry. All right, and that's a really important metric because, like, for example, under drought stress, those photosystems start to break down. Remember, we talked about that all of the enzymes in the electron transport chain, those are in the membrane, the thylakoid membrane. And as that membrane starts to heat up, it starts to deform. And then all of those enzymes lose some of their functionality. They're not performing as well. And so if that's happening and those photosystems and those enzymes aren't working as well, more light is going to be fluoresced back out than absorbed. And so that will change, or sorry, more light will be unused in photochemistry, and so we'll get a much lower peak. And what that generally means is more of that light is being dissipated as heat. So changes in FV/FM are going to tell us the proportion of light used in photochemistry. And another way that this can be referred to is it's the maximum quantum efficiency of photosystem 2. Remember, we're only measuring photosystem 2, and so we use this symbol here. So phi photosystem 2 max for that maximum capacity. Because remember, that first pulse is when the leaf was dark adapted. So it was in the dark, the photosystem was completely shut down. The second metric we can get, oh, and I didn't put the equation here, is phi is of photosystem 2. Sorry. And what the quantum yield of photosystem 2, or phi of photosystem 2 is the difference, is basically the difference between those two peaks. All right. So the bigger the difference, the more efficient the photosystem is. Because remember, this is the fluorescence when all of those photosystems are closed. And this is once photosystem has turned on. So if a plant's really healthy, we would expect this peak here to be really low because it's using all of the energy, and it's all of the photosystems are open and ready to absorb more light. So basically that difference between those peaks is what's driving that quantum efficiency of photosystem 2. And then with that number, with the photosystem 2 quantum efficiency, we can calculate the electron transport rate. And remember, this is something we could estimate from an ACI curve. So we could get Jmax, which was the maximum rate of electron transport, from ACI curves. We can get that same parameter from chlorophyll fluorescence. And this is a little more of a direct measurement, because in this one, we're actually stressing out or kind of triggering the light reactions to work and in measuring exactly what those light reactions are doing. So this is a little more direct measure of electron transport rate, but we can get it from both of those types of measurements. And so that's just the equation we would use to get that. So what we're going to do next Wednesday is we're going to look at a couple fluorescence curves and kind of estimate FV and FM from those curves and calculate these rates for some species, different species, and then kind of think about what that actually means. So, like, don't worry too much of all of the details of the fluorescence right now. I want you to be familiar with it, kind of know what the process is and how it's like triggering photosystem 2 and fluorescing or using that light in photochemistry or producing it as heat. And just so have a general idea of the mechanics of this measurement, and then we'll look at some data and hopefully it will make more sense. So why would you measure chlorophyll fluorescence? Well, as I pointed out, it's a really fast measurement, and it's a relatively easy way to estimate the level of stress a plant is experiencing. Because what you can do is you can compare, the most common thing people compare is FV/FM under different situations. So remember, FV/FM is like the maximum capacity for converting light to photochemistry. And so if that goes down in response to drought, you know that the photosystems and all of that machinery are starting to break down and they're not operating at their optimal rate. And so that gives you a metric of how stressed a plant might be. So easy way to measure measure leaf stress, and also that a lot of times that stress is coming from photoinhibition. It gives you some detailed insight into photochemistry, how fast those processes are happening, how many photosystems there actually are in particular leaves. And because this can happen over relatively short time scales, you can look at like diurnal patterns of stress. And so plants can go through a pattern where they're relatively healthy in the morning, they get stressed and those photosystems start breaking down in the middle of the day, and then they can repair themselves and be healthy again at night. So you can see kind of what's going on over the course of the day. As I mentioned before, it's much faster than a gas exchange system. It's really easy to do in the field. The equipment's lighter, the batteries last longer, all that kind of stuff makes it a bit easier. And then I think I also mentioned this earlier on, the equipment tends to be a bit cheaper. A good fluorometer is in the ballpark of, I'm trying to remember the last quote I got, but somewhere around 10-ish grand to get a fluorometer. And there are some cheaper ones out there that have limited capabilities but are still useful. That might be more like 5 grand. I'm just ballparking numbers. I don't remember the exact ones. But clearly cheaper than a 6400, which is more like $50,000. Okay, just a couple of figures to show kind of how these parameters relate to other measurements and how they might be used for understanding plant stress. In this figure here, we have that FV-FM metric. So remember, that's the proportion of light absorbed by chlorophyll that's going to photochemistry. So this is an efficiency metrics. And this is quantum yield estimated from a light response curve. And you can see there's a really high correlation between those two metrics. So they're telling us, similar pieces of information about how efficient the photosystem is at absorbing and using sunlight. Another way we can use it, as I mentioned, is ignore these exact equations. They're a tiny bit different than the ones I showed you before. But the other metric we can get is the electron transport rate. So you can see in these two curves, on the x-axis we have irradiance. So this is much like a light response curve, but instead of photosynthesis on this bottom panel, we're measuring electron transport rate. But then if you look at the light response curve from this plant, so this solid line here is photosynthesis measured with a 6400, you can see that the electron transport rate is matching the photosynthetic rate really quickly. So you can estimate electron transport and then start generating light response curves based on that metric instead of actually measuring CO2 uptake. And so it's much faster at getting that same type of information. And then here's a figure from a paper. There's a link to it if you want to check it out. So this is a bunch of different leaves that have been exposed to a disease. And so you can see there's on the far left, we have healthy leaves. Then we have some that are infected with the disease and then some that are just nutrient deficient. And what's plotted, if we just look at, they're not in like a column, unfortunately. But we look at FV-FM up here. Let's just focus on this top left panel. You can see there's a few FV-FM metrics, but we'll just focus on this one up here. So FV-FM in a healthy leaf is really high. That means the difference between that maximum peak and its baseline is really big, which tells us again that those photosystems there's a lot of photosystems that are operational. But you can see when it's infected with the disease, this disease is having some impact on the photosynthetic machinery of the leaves. And so there's some places in this leaf that have relatively high FV-FM, but lots of places that have much lower FV-FM values, which again means those photosystems are not working anymore, and so they're not even fluorescent light when they're closed. And all these images were taken with a hyperspectral camera, so you can get this really high resolution of what's going on an individual leaf. So just another kind of way that fluorescence can be used to understand how different conditions are impacting the functioning of a leaf. Here's one where in this example we have two species that were exposed to drought. And so the closed circles are 1 day after drought, and then open circles are 4 days, and then 5 and 6 days after drought. And you can see they generated a light response curve measuring the electron transport rate at different light intensities. And what's cool about this is here's one day after a drought, so we have, it's a relatively healthy leaf, still photosynthesizing well. And then a couple days after drought, it really drops, but then it's able to recover. And so day 5 and 6 are closer to that original electron transport rate at high light intensities. And on this species, we've got, relatively high rates. And then day 4, it drops a bit. Day 5 drops more and then rebounds by day 6. So the ability to kind of recover and deal with drought of these two species happens at different time scales, which is kind of interesting. So that's another way that it's been used. And then one more way, if I can get, oh no, there we go. Okay. Measuring, so in this case, this was a study where we grew 4 different species under 2 different CO2 concentrations, and then exposed some of them to drought and some they were well watered. And so you can see those symbols and colors over here. So ambient is the circles, and then elevated is triangles. And then in this case, we used the fluorometer to measure quantum yield at different light intensities, the par values here. So you can see, let's see, this is, oh gosh, I'm going to blank on the names. It's a ruse. I can't remember that common name. And this is juniper. This is Utah juniper. We'll focus on that one. How about that? So under ambient conditions and droughted conditions, we see a drop in quantum yield. And so quantum yield is going to be related to how big that peak is relative to baseline. But under all other conditions, the quantum yield is pretty high. What's interesting about this particular species is ambient CO2 and droughted, we have relatively low quantum yield. But when you drought a juniper but elevate CO2, it actually compensates for that droughted effect, and our quantum yield is a bit higher. But if you look at these species, you can see very different reactions. Some of them are driven primarily by ambient versus elevated CO2, and then this juniper has an interaction between CO2 and drought. So some cool things you can tease apart about the health and operation of plants under different conditions. All right, so just a summary of fluorescence. It's a way to estimate the photosynthetic performance of plants. And the metrics we get from fluorescence the metrics we get from fluorescence are correlated with photosynthesis, but they can differ. So for example, photorespiration rates, if those are high in a plant, it'll be using the energy from the photosystems and not having it fluoresce. And so that would make a plant look, from fluorescence measurement, we would say, hey, this plant is performing quite well. But it might be photorespiring really high. So if we measured CO2 assimilation, we'd get a different answer than if we measure chlorophyll fluorescence. So that there's not super common that there's those exceptions, but it does happen. And so you do have to be a little careful in your interpretation using fluorescence. And then the other thing that is important to know about it, I guess this isn't really a summary, but just some important notes, is that you can really only tell the relative difference in performance. So because the baseline value in that F max so you need to have the F sub 0 and F max of an individual leaf before you can tell what the fluorescence is once photosystems are up and operational. And so you have to have all of those measurements on the same leaf in order to make anything of the data. Just collecting fluorescence on an individual leaf doesn't really tell you how fast it's photosynthesizing or how healthy it is. You have to have measurements over time and then compare them. So it's a relative metric, is what I'm trying to say, and I don't think I did a good job. But just remember, it's relative, and so you need to, if you're going to measure fluorescence in response to some environmental stress, you have to have some healthy plants to compare it to your unhealthy plants. So that's what I was trying to say. Okay, we're done for today, and we'll work on a little bit of fluorescence data next week.